There.
Looks good to me.
All right. Well, thank you everyone for all those awesome photos and for presenting the data. I'm new to the area, so it's fun to see this community and to get to know you virtually at least. Thanks for inviting me to give a little guest talk here. I'll just jump right into it. So yeah, I'm Mike Johns. I'm a researcher with Point Blue Conservation Science, which is a nonprofit based out of California, which some of you may have known. It's formerly known as Point Reyes Bird Observatory. So we went through a name change in 2012. But I'm going to talk to you about the pigeon guillemots that we have on the Farallon Islands, some historic trends that we've discovered, and then some new insights we're gaining from some of the tagging work we've been doing. So, contents of the talk, I'll just, like I said, pretty much all like I just said, introduce the Farallons, talk about some long-term monitoring work, and then talk about some tagging studies. So Southeast Farallon Island is located approximately 26 miles off the coast of San Francisco. So it's actually an archipelago of roughly 3 islands. There's North Farallon, Middle Farallon, and South Farallon. Southeast Farallon is by far the largest of the group, and we call it SEFI for short. It's part of the Farallon Islands National Wildlife Refuge, so it's managed by the U.S. Fish and Wildlife Service, and Point Blue is simply tasked with conducting the research on the island. So this is a photo of the Farallons during the summer. What it looks like, all the vegetation is now gone. It's very dry, and we've got a humpback whale fluking up in the foreground for good measure. This is what it looks like in the summer, or in the winter, sorry, with the Farallon weed, an endemic plant to the island that covers most of the available territory. So the Farallons are located right along the edge of the continental shelf break, and it makes it a really productive region. We have some really persistent winds in the spring, some northwest winds that force the surface water offshore, and that water is replaced by very deep nutrient-rich water. And when those nutrients hit the surface, they ignite these phytoplankton blooms. We get a lot of productivity happening. That works its way up to things like copepods and krill, and eventually the marine predators like seabirds and whales. So here we have a humpback again, gorging itself on some anchovy. And just a bit of history on the island. So the Farallons were occupied sometime in the mid 1800s. There were these things called the egg wars that happened out there. So San Francisco was lacking in chickens, and they harvested murre eggs from the island to supply all the bakers and needs of the city. So a lighthouse was established on the island in 1855, and that's when these dwellings were built to house the lighthouse keepers' families. And these are the same buildings that we use today. We've renovated these houses, and we now use them as a field site. And yeah, Point Blue established a field station in 1968, so a pretty long running time on the island. And we've contained a continuous presence on the island ever since. So we have personnel and staff that are living on the island year round. We break it up into 3 different seasons. So we have a winter pinniped monitoring season, a summer seabird season, and then a fall landbird migration study. So constant presence since 1968, pretty incredible for a nonprofit. It's difficult to get on the island. I love this photo of this woman being lifted on a chair from a supply vessel, and she's going to be dropped on a skiff taken out to be dropped off on the island. We have a similar setup. We don't use chairs anymore, but here we are launching a Zodiac into one of the gulches to take out to one of the supply vessels. So we have a fixed crane on the island that we use to do this. Okay, so getting into the seabirds. So there's roughly 13 species of seabirds that we monitor out there. This is just a snapshot of the core species that we follow. So we've got tufted puffins, Brandt's cormorants, rhinoceros auklet, western gull, Cassin's auklet, common murre, ashy storm petrel, and then our beloved pigeon guillemot down here in the lower right. And the projects have been pretty consistent throughout the years. The methods have stayed the same. So here's a photo of an anonymous researcher in 1968 weighing a Caston's auklet over a bucket with the Piscola scale. And here we are present day, basically doing the same thing. So over 54 years now of continuous seabird monitoring data. And in terms of the pigeon guillemots, which we call pigus, so here we are, here's their range on the Eastern Pacific from Southern California up to the Bering Strait and along the Aleutian Islands, and this red circle is the Farallons. So on the Farallons, the pigus breed mostly on the steep, sort of deteriorating granite slopes. There's a lot of boulders and crevices for them to tuck underneath, lots of cracks in the granite that they utilize. They compete with Caston's out there, so there's roughly 30,000 Caston's auklets that breed a bit earlier than the pigus do, and so there's a bit of overlap in terms of breeding habitat between those two species. Recently, there's probably about 3,000 breeding individuals on this very small island, so it's a pretty busy place in the summer. You hear the pigu calls basically everywhere. It's a constant chorus throughout the breeding season, so it's pretty fun for pigu lovers. It's a pretty awesome place to be. They're everywhere. The only predator they have on the island are the western gulls, so it's the largest population of western gulls. There's probably 12,000 western gulls on the island, and so they will definitely kill whatever enters their territory. And when the fledglings are leaving in the fall, a lot of them will get picked off by those western gulls. So getting into the details of the monitoring, so we follow roughly 90 crevices and artificial nest boxes each season for breeding success and for timing. To track the population, we simply go up to the lighthouse, which is about 300 feet high, and then with the scope, just scan the surrounding waters and count all the large rafts of guillemots that form in the spring. And then we have our diet studies. So you guys are all familiar with this. We're just tracking birds as they fly in with a bill load of fish or a single fish, and then we're identifying its, IDing it and getting a size estimate as well. In terms of the monitoring for reproduction, so that happens between April and September, which is their breeding season out there. This is a photo of some of our assistants scrambling up Lighthouse Hill. We spray paint or hand paint all the crevices that we follow with numbers, and then we scan these sites during chick provisioning to get at diet as well. So they're just going around and checking these breeding sites. They're checked every 5 days in the afternoon to avoid adult disturbance. The adults tend to not be present in the afternoon when they first lay their eggs. We weigh the chicks every 5 days to track growth throughout the chick growth period. And then when the chicks reach mostly feathered, we'll put a metal band on their right tarsus. So we want to track longevity and also maybe get recovery rates from birds that are found dead in other places. So just looking at some of the data, I do have some graphs to show you. So this is just our long-term data set for mean number of chicks that are fledged per pair. And you can see, which is a good sign, over the past 50 or so years, we don't really see much of a trend in breeding success, either up or down. So it's good to see the population's not declining in some way in terms of breeding success. So this just shows the annual variability. It kind of hovers around 0.8 chicks per pair. As we know, these pigeon guillemots can lay up to 2 eggs, and generally that second chick is more of an insurance policy. So on a really good year, they might fledge 2 chicks, but generally that second chick ends up dying. So some of the poor years are noteworthy, like we had the 97, 98 El Nino where we had pretty low productivity, and then this 2005 through 2007 warm event where we had some pretty low productivity too. So that's highlighted in the breeding data. Another complicated figure, I suppose, but this is the median lay date for the first egg over time. That's this black line here. And then the colored tiles are just showing you the total number of pairs that laid that first egg in this date. So if you have a warmer tile, that means there's more birds that laid their eggs during that period. And I show this figure just to say that in the last couple of years, we had the earliest lay dates on record. earlier than we've ever seen before, and there's some internal debate as to whether or not this is going to be a long-term trend or not. So stay tuned for the next 50 years. We'll see if they start breeding earlier on the island. And this is just our annual raft count. So this is the high count for rafts around the island in the spring, and we use this as a proxy for the population size. So you can see it got up to nearly 5,000 birds in the mid 2000s, and then it's been or around 2014, and it's kind of slowly dropped, but this is probably just a lot of observer error and that sort of thing. So it's hard to say if the population is actually declining currently, but it's most likely remain stable around 3,000 birds. Sorry if my mouse is making weird sounds. I'm trying to stop that with water. Okay, and then chick provisioning diet. So we do these surveys every morning, every 3 days when they're provisioning their chicks from 7 to 11. So we're tracking the birds as they fly in with binoculars, and we're noting while in flight the species, the size of that prey, if the bird's banded or not, and then what site it goes to. So that way we can link diet information with breeding success of these followed sites. So we usually have 2 observers watching the birds come in and then 1 data recorder. This is just a group outing in this photo of people being trained. And the resolution's a bit poor on this, but this is our annual proportion of chick diet throughout the years. And similar to what you all see here, sculpin are showcased quite a bit in their diets. But generally, these birds are targeting juvenile rockfish. It's one of the most important prey items for seabirds on the Farallons. We see that in the murre diet and the cormorant diet and the auklet diet and the pigus as well. And when the rockfish aren't around, they're going after mostly flatfish and sculpin. They take gunnel and brotula and things like that occasionally, but those are the 3 main prey types we see. And this is just a pitch for any potential, if you know any grad students or potential grad students out there, it's a low-hanging fruit, just trying to figure out how important rockfish is to the diet of these pigeon guillemots, this population. There's a pretty strong relationship between rockfish and sculpin. So when, like I said, when rockfish aren't around, they're targeting sculpin instead. Rockfish are probably more energetically important to them than sculpin are. So anyways, if you know any budding grad students out there, let me know. Okay, so in the new studies, I'll just touch base on some of these new results we've got from the tagging work we've been doing. And I think Scott Pearson may have presented some of this stuff last year, but so this might be a repeat a little bit. But so there's been a lot of anecdotal evidence that our population comes up here in the winter time. We have a few band recoveries from dead animals in Washington State and British Columbia. There's a pulse of migrating birds that appear off the Washington coast in the spring and fall. So, and there's an absence of guillemots in California during the non-breeding period. So it's always been assumed that they do this migration, but we wanted to actually show it with tags. So this is a photo on the right of a cassins wearing what are called geolocators. It's attached to a plastic band that we then adhere to the bird's tarsus or wrap around the bird's tarsus. I don't have a photo of a guillemot, so this will have to suffice. This will do. So these devices, they record ambient light level. They have a battery life that lasts up to 2 years. But the sad thing is they have to be retrieved to get the data back. So that makes it a challenge. And we deployed a total of 90 devices between 2017 and 2019, so 30 tags per year. And we're able to recover 67 of those 90, which I think is a success. Not bad for a tagging study. The main reason that the success was not higher was because of the Western gulls. So for those of you who are familiar with Western gulls, they're very aggressive and they're very loud. And when you have 13,000 of them, basically sneaking up on a pigeon guillemot is virtually impossible because they sound the alarm every time we approach. So the guillemots are cued in on the gulls. They know if there's danger around and they'll flee their crevices if they hear us coming. So Western gulls are always a problem. Here's just a quick anatomy of what a geolocator is. So we've got a memory chip. Here we have a little light sensor, a battery pack underneath, and these 2 little metal prongs, which are used to both detect whether or not the tag is submerged in water, and also you can hook up a cable to these to uplink to the computer. And this one actually had one of the tags we recovered had a barnacle growing on it, which shows you how much time these guillemots are spending in the water during the winter time. Pretty much all of their time, essentially. And a quick schematic on how this works. So the geolocators, it's not like a GPS unit. It doesn't just give us a latitude and longitude. It's just passively recording the light values that the tag's exposed to. So this cartoon bird is, it's no light right here. It's experiencing a sunrise event. So the light values jump up, it goes about its day, the sun goes back down. And so we can use basically the timing of these twilight events, these sunrise and sunset events, to estimate roughly where the bird is on the globe. So when those events happen and how long they're spaced apart will tell you latitude and longitude. And I won't go into detail about that. I had some slides, but I took those out. Just if you have any questions, you can ask later. But so here's the results from that study. So basically, these are called kernel density plots. So if you see warmer colors, that means there are more points in that area. The cooler colors are fewer points. So in September, we see the birds are departing Cepheid here and flying basically on a migration along the coastline up to Haida Gwaii. In November, we see they're leaving Haida Gwaii and finding some residency locations in Southeast Alaska and British Columbia, and they're still there in January. And by March, most of the tag birds have departed the area and have begun their migration south again back to the Farallons. And I think you may have seen an animation of this that's somewhere along the lines as well. I didn't include that, but so that's their annual cycle. Pretty cool. We didn't know this before this study, so it's pretty exciting to get some actual conclusive evidence of their migration. This breaks it down in a different way per year. So just walk you through these plots. The bubbles, they're colored by the month that the bird arrived at that location. And then the size of the bubble indicates roughly how many days they spent there. So a bigger bubble is more days spent at that spot. And then a purple bubble, for example, is a bird that arrived in October. Actually, I can't see the legend because this thing's blocking me, but I think that's right. And we found that they had 2 stationary periods during their non-breeding period on average. And the first stationary period, they arrived there sometime in early September and stayed for roughly 45 days. And those tended to be around Haida Gwaii. The second stationary period they usually arrived around November and then stayed for roughly 131 days. So that's their longer winter residency locations further north in northern BC and Southeast Alaska. So a couple more graphs. So another way to look at this is we wanted to look at their timing along the migration. So let me just go back here. These A, B, C, D, I call these benchmark latitudes that we were interested in. So those are reflected down here. This is Cepheid, Puget Sound, Queen Charlotte Sound entrance, Dixon entrance, and Admiralty Island. So these are increasing latitudes as you go up. And broken up they're colored by the tagging year. So you can see the birds are leaving the Farallons roughly around August 26th on average. And they're making this pretty much continuous flight until they get just past Queen Charlotte Sound. And then there's a gap before they arrive at Dixon entrance. So that's that first stationary period that's happening sometime in late September, early October. And that happens to overlap when we assume these birds are going through their pre-basic molt. So when they're molting all of their flight feathers and they're rendered flightless for a brief period of time. So it seems that this location around Haida Gwaii is an important molting area for this population. And for those of you who are familiar with the area, which you probably are because you're around here, Hecate Strait, I think is how you pronounce it, is a pretty shallow region that might provide some good foraging habitat for these birds, where they can still be far enough away from shore to avoid shore-based predators, but still have access to shallow benthic foraging. So yeah, there's a longer delay that happens during their fall migration north, and in the spring, it seems like they're making pretty much a continuous flight back to the island. So they're not really stopping much on their way south once they wrap up their pre-alternate molt, when they're coming back into breeding plumage. So yeah, so that fall migration takes about 49 days on average, and the spring migration takes about 24 days. So much quicker coming back. They're excited to get back to breeding, it seems. And Maria, I think you're on the phone somewhere. You sent me an email asking, you know, if we know roughly how long they're flying per day. And I didn't really dive into that too much. I know that the total migration is roughly around 2,400 kilometers. So that you can do some maths there to figure that out. And we set these tags to record wet dry data too, to get at sort of behavior along the migration. Again, I'll walk you through this plot. So this is colored by the proportion of the birds that were completely dry during a 1 hour period. So they never interacted with the water. So if it's really red, that means all of the birds that we tagged didn't land or touch water during this hour chunk throughout the day. And so what this graph is telling you, and these vertical lines are saying when roughly the population has fledged all their chicks, and then on the other side in June, when roughly most of the birds have laid their eggs, when breeding has started happening. So you can see during the breeding season, these birds are spending almost all of their time on land during the night. So they're roosting at their sites or potentially brooding or incubating their eggs. And then during the day, during the breeding season, they spend some of their time on the water, but once migration occurs, or once they're in their winter residency areas, they're virtually not on land at all. They're pretty much in the water constantly. And the question about how often they spend flying throughout the day. So we do see these periods, particularly in the spring, when birds are traveling back south, where we have some instances where they're completely out of the water, or some of the birds are actually completely out of the water for an hour period. So these likely represent that intense migration south that's occurring sometime in late March, these little spikes that you're seeing here. There's a lot to unpack with this figure. That's sort of a snapshot of it, but basically what it's saying is that so yeah, during the breeding season, during the dark hours, they're on land, and then these periods where they're dry during the non-breeding season at night are actually likely when they're just resting on the water. So it's been shown in other studies where things like puffins will actually tuck their legs up into their body when they're resting on the water, and so you actually get a false reading of dry because they're so good at keeping their legs dry when they're sleeping on the water. So these periods at night that are dry are likely birds that are just sleeping on the surface of the water. And this work was mostly, so we've been also tagging Cassin's auklets and rhinoceros auklets as well from this population. And you can see these two figures here, the pigeon guillemots are doing something completely different than the other alcids at this colony. So Cassins are remaining pretty close to the Farallons throughout the winter. Rhinoceros auklets tend to move south a bit towards Southern California and Baja. So this is pretty, interesting behavior that these guillemots are exhibiting. And it also shows that they're going to face different threats during the winter months compared to some of these other species. So this work was actually funded to get at how risky their winter movements are, put them in terms of potential risk to being exposed to oil spills. So, the Farallon Islands is located pretty close to a major shipping port. Lots of vessel traffic comes in and out of San Francisco Bay. So we know they're highly vulnerable to potential oil spills in the summer. And these new tagging data are telling us that potentially, different species are going to have different exposure risks during those non-breeding periods. So we're trying to develop a predictive model to look at using vessel tracking data and proximity to major ports. What are the risks? What's the potential oil spill risk along the coast? And then overlap that with these seabird distributions to get at how vulnerable they are to a future oil spill. So that work is ongoing. And I kind of have to mention that because that's why this project was funded, but we really just wanted to look at what they did in the winter. So, okay, and finally, one last thing, we're starting to put GPS tags for summer foraging trip data. So these are actually just taped to the back feathers of the birds, and they only stay on for maybe 5 days. Again, this is a Cassins on the right, but we have a Pigeon here on the left wearing one of our GPS tags. And this is a wooden nest box that we monitor. And these are great because they actually can upload their data to a UHF, through UHF to a base station, and we could just download them remotely. So we don't actually have to get the tag back. So it's actually pretty difficult to recapture these birds once the chicks are old enough to be left alone. So they're not spending much time at the site. And just really quickly, these are some pilot data for that. These are just simple tracks. So far, we put out 6 tags this summer, and we got 5 to collect data, and 3 were actually recovered, which is pretty cool. And these are just showing roughly where they're going when foraging for their chicks, and it shows that they're staying. Each one of these concentric circles is 2 kilometers, so it's showing that they're staying pretty close within 8 kilometers of the island when they're provisioning their chicks. And this is all preliminary, so there's a lot more to tease apart with these data. But I just wanted to let you all know that it's happening. Yeah, so what else can the PGs tell us? These are some future questions that we'd like to explore. We want to look at coupling our diet watches with our GPS tag data to see if we can figure out where they're finding the sculpin, where they're finding the juvenile rockfish, identifying any important hotspots around the island that might need protecting. And then, we'd really love to collaborate with other groups throughout their range to deploy geolocators and GPS tags to do more of a range-wide comparative study about, are our birds on Cepheid representative of migratory behavior, or is it a very special behavior only shown in this, particular region in California? Okay, that's all I have. Hopefully that was informative, maybe not too long. So thanks again, you guys, for inviting me to join your group. And I'm in the area, and if you need help with surveys, I'm gonna have to reach out because I also like PDUs. So thanks again.
Thanks so much, Mike. That was excellent. Really, really exciting stuff. I imagine that there's going to be a few questions. Hopefully you're up for answering a couple.
Yeah.
All right. Folks, I didn't really go over what the best way to do this is. I think it's totally legitimate to just unmute yourself and ask a question, but if you want to type a question into the chat too, I'm happy to ask, or Mike can probably look at it too, either way. Anyone have anything? I have a question. You said raft counts. I'm just assuming it's literally a count of the birds that are rafted up around the island. Is that right?
Yeah. So they tend to stay within maybe a mile or so in the spring. They all kind of gather in these big groups around the island. And so we just basically with binoculars and scope just kind of scan 360 with a clicker and just count all the birds that we see. Yeah.
Great.
One thing I did mention was we used to do evening counts, which produce much lower counts than our recent morning counts, which we started, I think, in 2000-ish or so. So, yeah.
Awesome.
Hey, Mike. Mike, this is Govinda from Whidbey Island. Great presentation, very informative. The question that I have for you is, and maybe you'd mentioned this and I had missed it. I'm also trying to kind of cook dinner a little bit, but geolocators that you did on the Paigus, what was your sample size on those fancy little thingamajigs?
Yeah, we did 3 years of tagging and 30 tags per year, so a total of 90. And they run at about, I think it's like $120 these days. So they're actually pretty inexpensive. We had a pretty small budget. So geolocators are a good fit for small budgets.
Cool. And then maybe just a follow-up question. It was interesting that you guys were painting or marking the burrows. And I wonder if that would alert any predators that like, hey, Paigu is right here, or is it just because the gulls are the predators and they're just dumb enough that they, they're just easy pickings when the chicks come out or whatever?
Yeah, that's a good question. I don't think we ever actually looked at, I mean, the markings are usually, they're not necessarily on top of the crevice. Sometimes it's like an arrow pointing to where the crevice is. And there's quite a few markings on the hillside. So I'm not sure a predator would be able to cue in on that pattern. And I did forget to mention that we do have peregrine falcons on the island, which will take adults as well. I didn't say that. And often, I don't know if this is anecdotal, but well, it is anecdotal, but we had, I think, 3 of our geolocator birds were found dead, killed by peregrine falcons. So perhaps the jewelry that we're putting on their legs, they could potentially cue in on that as well. So
Gotcha. Thank you.
Yeah.
Awesome. Thank you.
There's a few questions, also a comment from Terrence from Nisqually Reach Nature Center. He was talking about Dr. Seraph Converse, who's one of our collaborators, one of the people that's really helped with our work, is doing that on Protection Island, is using these, they're GPS tags. So similar situation. And people were asking about using geolocators with pigeon guillemots in the Salish Sea. It's not really something that's cooked into our protocol, and not necessarily something that we'd be doing, as part of the breeding bird survey. There's certainly room for it, and it would certainly answer lots of questions. I think that, I think Sarah might have some thoughts on this in tandem with our work. What I will say is that if people are interested in hearing about what she's up to, she did a presentation for us, I think it was in August, and I have a recording of it. I haven't posted it online because she asked us not to just publicly post it. But if you email me, if folks email me or get in touch, I'm happy to share that. Sorry, Mike, that's not really a question, but.
Oh yeah, thanks for pointing. And we have been talking to Sarah, and we may be collaborating on a project next summer. I actually have a batch of these devices in my fridge staying cool, so we might bring those out to Protection and repeat this study on Protection. Yeah. One thing that we found too with this work, not too many people have handled adult guillemots for a tagging study, and we've demonstrated that it has pretty much no effect on the hatching success or breeding success of the birds that we followed. So that was a good, reassuring finding that our handling wasn't too disturbing to them. So.
Mike, I have a question about the connectivity of these populations along the West Coast. We know that there are birds that winter in the Vancouver area and Vancouver Island, and we know that our birds disappear pretty much. Do you have any thoughts on the prevalence of pigeon guillemots from other areas, like from the Salish Sea, along the Southeast Alaska coast or North BC, besides the ones that you guys have tagged and followed?
Yeah, well, there's really no way to confirm that yet. It's assumed, so there's scattered colonies throughout California, not just on the Farallons, and it's assumed that those birds likely make a northern migration as well. And there have been a few kind of older studies that looked at wing morphology, and it showed that birds on the extreme ends of the range, so birds that are up in Alaska and then in Southern California, tend to have longer wings than birds that are breeding in sort of British Columbia and the more contracted part of their range. And so it could be that birds on the extremes have more of a migratory tendency than birds that are breeding sort of around the BC area.
Right.
But yeah, it's hard to say. I mean, it's so hard to get to some of these colonies. So yeah. as you all know.
I have a question about people. Maybe I missed it. How many folks are out doing, I guess, because I know there's all sorts of stuff going on there. But on average, what would you say, how many people are on the island doing work throughout a year?
Yeah, so in the seabird season, we start out with 3 people, and then that ramps up to a total of 8 in July. So that's when we have basically all of the species are on eggs or have chicks. So it gets pretty busy. And then in the fall and winter, yeah, the teams are roughly 5 to 7 people. And that's just our Point Blue staff. We also have the Fish and Wildlife Service has a presence occasionally out there to do maintenance work, and we have contractors that come out to do various projects. So it can expand to roughly 20 people, but it generally around 5 to 8.
Cool, thank you. Puts it into perspective, having a lot of birds in kind of a captive audience in one place versus the way that we do things inherently.
Right.
Very, very different. Anybody else have any questions?
Well, Mike, I have another question, only that I've heard about the rat problem on the islands. Is that still an issue, and that it's kind of like a carpet?
That's a big topic, the house mouse eradication project. Yeah. We've passed some major legislative hurdles, the California Coastal Commission has given us the green light, or at least the company we've contracted to do it. So potentially in the next couple of years, that eradication will happen. But I don't know much about the details of it. I just know it's that's a hot button issue in the Bay Area. So yeah.
Thanks.
But it's like the largest concentration of house mice in the fall. The ground is just literally moving with mice. It's insane.
That's what I've heard.
There. Yeah.
I have a question, Mike.
Mm-hmm.
Your survey site is very much, like you said, a granite rock out there in the water, and a lot of the areas up and down the coast are more coastal. And here we are in Puget Sound with our erosional bluffs. Do you know of other areas that would be similar to our geologic setting here in Puget Sound area that where they mainly do breed in the bluffs?
In this area?
No, I mean within the range.
Mostly the Farallons are actually kind of unique in this offshore island setting. Most of the breeding range-wide occurs on the mainland in these coastal bluff regions. Like there's a decent colony in Santa Barbara that's on a highly erodible bluff, and I don't know too much about the Alaska populations, but I'm assuming they're mostly in sort of the rack that occurs along the shoreline. But so the granite, the setting that we have in the Farallons is definitely, it's special in terms of habitat for them. I think it's definitely different.
Yeah, and they do like areas around like Deception Pass here on Whidbey Island, where it's very rocky, and
right.
A lot of offshore rocks that you see off the Washington, Oregon coast have them. So
right.
We also have been watching ferry docks and other structures where they manage to figure out some way to breed up and under. It's very fun.
There's a story of the colony on Alcatraz in San Francisco. There's a pair that breeds on a crane boom that goes up and down to load gear on one of the boats. So it actually rides the boom. It's basically anywhere they can find, I guess. Yeah.
And we've watched them ride the ferries here. And I remember going to Haida Gwaii and taking one of the little ferries across there, and the skipper of the ferry was telling me that they breed in the ferry and go back.
That's wild.
Yeah.
Yeah.
All right. Well, Mike, thanks so much for joining us tonight. This is really, really lovely. Really awesome presentation. Really great to hear about all the work you're doing. Thank you for doing all the work too. Yeah,
It's also fun.
it's excellent. Yeah.
Yeah.
And welcome to the Pacific Northwest. Hopefully we'll get you out on the beach. Get in touch if you want to, join in on the survey. There's not any surveys happening, around Tacoma. Vashon and South Sound kind of, bookend the region. So
All right. Well, maybe I found my niche.
yeah, there we go. Anyway, and thank all of you again for being here. This is what makes this work so special, is having all of you here. So really appreciate it. And thanks again for the great season. I'm going to go ahead and stop recording. As always, you can email questions. guillemotresearch@gmail.com is the email address that I use as the coordinator. So that's always an easy place to send questions. But the folks who manage your surveys as well can always get in touch. And we will call it that.
Thank you.